Welcome to F-Block. This is my cell communication project on SARS-CoV-2 spike protein epitopes, better known as COVID-19. So before we actually get to signal transduction pathway, we have to talk about how we get the ligand. And the ligand will happen through the binding of COVID cells to the host cell. This can either happen through endocytosis when it binds to ACE2 or when it binds and fuses through the membrane. We can see here. So here we can see where the COVID cell will enter into the host cell, and through a process of uncoding will have its raw RNA. This RNA will be processed, and this RNA will then go and code in the rough endoplasmic reticulum for more viruses to be created. Now, we're going to be focusing on the spike protein because it's the most well-researched, and it has the best antigenic properties. So what will happen is sometimes when these spike proteins are being replicated, instead of going into a virus, they will be broken down by something called proteases, which we can see here. These proteases will break them down into individual peptides, and these peptides will be attracted to something called the MHC1 complex. When these peptides are attracted to the MHC1 complex, they will bind and create an MHC1P complex or an MHC1 peptide complex. This is when they will start to be called epitopes, as they will move to the cell membrane and attract something called TCR on CD8 plus T cells. Now, the TCR is a non-catalytic tyrosine receptor, and it will bind to the PMC1 through juxtatyne signaling. As we can see here, it'll come and it'll bind, and at the same time, something called a co-receptor, which is CD8, will bind to the MHC1 complex, and as it changes shape, it will cause the activation of LCK, which is a kinase, and as this kinase gets activated, it will go and activate ZAP70. Now, this ZAP70 is going to go and get recruited by the LAT signalsome, creating the LAT multiprotein. This LAT protein is going to go and it's going to activate the RAS-G protein. When this RAS-G protein gets activated, it's going to go and activate the RAF kinase. This RAF will start a phosphorylation cascade, starting with RAF, going to MEK, and ending with ERK. Now, this ERK will come and it will phosphorylate AP1, which is a transcription factor. It will go and code for 3 different molecules. We'll have granzymes, perforins, and then a bunch of cytokines, including TNFA. These cytokines, which I don't really go into, will cause inflammation, leading to more immune cells coming to infected places and T cells dividing quicker. Here we have a granzyme, and as we can see, it's going to enter through the perforin pore that is made. As this granzyme enters through the perforin, it's going to go and activate something called capsases. These capsases will go, and they will trigger apoptosis through another signaling pathway. And that was a lot. Thank you. The end.